Hello and welcome back to the Biochemistry for Health Sciences channel. In the last few videos, we talked about enzyme kinetics and how we determine two very important values. And these are Vmax and Km from curves such as hyperbolic curve and the sigmoidal curve, or from straight lines such as the Lineweaver-Burk plot. In today's video, we shall look at the applications of enzyme kinetics, and we specifically will talk about enzyme inhibition. Almost all reactions in the human body are catalyzed by enzymes. These could be reactions in metabolism, it could be reactions that make your blood clot, it could be a reaction that breaks down the blood clot. It could be a reaction that causes the muscle to contract, and so on. So there are hundreds and hundreds of chemical reactions that are occurring in our body all the time. And almost all of these reactions are catalyzed by enzymes. So for example, in metabolism, we have this very important metabolic pathway where glucose gets converted into pyruvate. This pathway is called glycolysis, and you can see it is made up of 10 reactions. And every one of these reactions, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, all of these reactions are catalyzed by specific enzymes. So these enzymes that catalyze reactions in the body, they are designed not only to make these reactions, to catalyze these reactions, make them go faster, they are also designed to regulate these reactions. That is, if the cell wants the reaction to go faster, it can regulate the enzyme, and the enzyme can make the reaction go faster. If the cell wants a reaction to go slower, then the enzyme is regulated so that the reaction goes slower. So by controlling or regulating one or more of these enzymes, we can regulate how fast glucose is going to be broken down into pyruvate. Similarly, we can regulate how fast blood is going to clot, or how fast the clot is going to be broken down, or how fast the muscle is going to contract, and so on. So almost everything in physiology that maintains homeostasis, we can regulate. So this regulation is extremely important for survival. Homeostasis, maintaining a fixed environment in the body in spite of changing environments outside, is very essential for our survival. The rates of enzyme catalyzed reactions can be changed by changing the Vmax and or the Km. The reaction will go faster if the Vmax is increased, and the reaction will go slower if the Vmax is decreased. Similarly, the reaction will go faster if the Km is decreased, and the reaction will go slower if the Km is increased. There are several ways by which enzymes can be regulated, and we'll look at these ways of regulating enzymes in future videos under metabolism. One of the most common ways by which enzymes are regulated is by the binding of small chemicals to the enzymes, and these chemicals are called activators or inhibitors. An activator is a chemical that will bind to the enzyme and it will make the enzyme catalyzed reaction go faster. And this can happen if the Vmax is increased and or the Km is decreased. An inhibitor, on the other hand, is a chemical that will bind to the enzyme and it will make the reaction go slower. And that can happen by decreasing the Vmax and or increasing the Km. So for example, let's consider the glycolysis pathway. There is an enzyme in the glycolysis pathway that catalyzes this reaction number 3. This enzyme is called phosphofructokinase, or PFK, phosphofructokinase. Now, ATP is a small chemical. When ATP binds to this enzyme, the Km increases. Therefore, ATP is an inhibitor. Increasing the Km would mean that this reaction, number 3 here, would go slower. This reaction rate decreases. And when this reaction rate decreases, the product of this reaction, fructose 1,6-bisphosphate, this product formation slows down. And that eventually slows down the whole metabolic pathway of glycolysis. So while ATP is an inhibitor, AMP is an activator. When AMP binds to this enzyme, the Km decreases. That will make this reaction go faster. This reaction number 3 goes faster. That means fructose 1,6-bisphosphate formation increases, and that will lead to increase in glycolysis. So since enzymes can be regulated by binding of small chemicals, this means that when we introduce foreign chemicals into the body, what we call xenobiotics are chemicals that are foreign to the body. For example, drugs and poisons. These foreign chemicals can also affect the catalytic functions of our enzymes. In fact, about 50% of drugs in the market today have been designed to inhibit specific enzymes in the body. Many poisons also affect enzymes. In this video, we shall focus on enzyme inhibition. In future videos, when we talk about metabolism, we will talk about enzyme activation. So remember, an inhibitor is a chemical that binds to the enzyme and it will make the reaction go slower, either by decreasing the Vmax and or increasing the Km. Inhibitors can bind to the enzyme by covalent bonds, or inhibitors can bind the enzyme non-covalently, that is by weak forces such as hydrogen bonding, ionic interactions, hydrophobic interactions. These are all non-covalent or weak forces. So inhibitors could bind to the enzyme either non-covalently or covalently. Inhibitors could be categorized as irreversible inhibitors or reversible inhibitors. So let's look at the difference between an irreversible inhibitor and a reversible inhibitor. An irreversible inhibitor is an inhibitor that binds the enzyme so strongly that it permanently blocks the enzyme function. This means when the irreversible inhibitor binds the enzyme is essentially dead. Now, the bond between an irreversible inhibitor and enzyme may be covalent, which is the more common situation, but it may also be non-covalent. Irreversible inhibition is not a common strategy that is used by the human body to regulate enzymes. However, it is very important in pharmacology when we deal with therapeutic drugs, and it's also very important in toxicology when we deal with poisons. A reversible inhibitor, on the other hand, is an inhibitor that binds weakly to the enzyme and temporarily diminishes the enzyme function. So it's not a permanent blockage of the enzyme function, it's only a temporary reduction of the enzyme function. So unlike an irreversible inhibitor, reversible inhibition leads to a temporary reduction of the enzyme function, but the enzyme function can return to normal once the inhibitor concentration decreases. Just like irreversible inhibitor, for reversible inhibition, the bond between the inhibitor and the enzyme once again may be covalent, but most commonly it is the weak forces, it is non-covalent. Reversible inhibition is a very important strategy that is used in normal physiology. Your body uses this to regulate your enzyme activity, and it's also very important in pharmacology when we deal with therapeutic drugs. Let's look at a few examples of irreversible inhibitors. So here, the first example we'll talk about are the organophosphorus insecticides. These organophosphorus insecticides, these are irreversible inhibitors of an enzyme called acetylcholine esterase. So at the active site of the enzyme, there is a hydroxyl group that belongs to serine, and this inhibitor will come and covalently bind to the serine hydroxyl group, and that would basically kill the enzyme. So let's see what happens if this acetylcholine esterase enzyme is inhibited in the human body. So in this diagram we have a neuron that is synapsing with a muscle cell. So this is a muscle cell, and here we have a neuron, and the two are synapsing, and this region here is called the neuromuscular junction. So when this neuron is stimulated, it will release this neurotransmitter called acetylcholine. It will release these neurotransmitters by a process called exocytosis, and these neurotransmitters, this acetylcholine, will go bind the receptor for acetylcholine present on the muscle cell. And when this acetylcholine binds the receptor, that will trigger an action potential. It will trigger a change in the electrical voltage of this muscle membrane, and that is an action potential. And this action potential will travel along the muscle membrane, and that will cause certain calcium transport proteins present in the muscle cell to open up, and when these proteins open, calcium is released into the cytoplasm of the muscle cell. Large amounts of calcium. And this calcium will then trigger the muscle to contract. Basically, the actin filaments will slide across these myosin filaments, and this muscle contracts, becomes shorter. So this enzyme acetylcholine esterase, it breaks down acetylcholine, and by doing that it makes certain that acetylcholine does not accumulate at the neuromuscular junction. So if this enzyme is inhibited, if this enzyme doesn't work anymore, then what will happen is the acetylcholine will accumulate at the neuromuscular junction, and the acetylcholine will continuously be bound to the receptor, and that means the muscle will be contracted. It cannot relax, it will always be contracted. And that can lead to serious problems, including paralysis, convulsions, and eventually death. In fact, this is how the insecticides kill insects and act as a poison in small amounts in case of humans. Another example of an irreversible inhibitor is the drug penicillin. Bacteria has an enzyme called transpeptidase. And this enzyme is very important for bacterial cells to make cell walls. So without strong cell walls, the bacterial cells cannot withstand the high internal pressure, and that will cause the bacterial cells to rupture. So penicillin is a drug that covalently binds transpeptidase. It's an irreversible inhibitor. It binds the transpeptidase, and this enzyme is basically dead. And without this enzyme, the bacteria cannot make the cell walls, and without their strong cell walls, they cannot withstand their high internal pressure, and that will cause the bacterial cells to rupture. And that makes penicillin a good, effective antibiotic. Another example of an irreversible inhibitor is the drug aspirin. Now, this drug aspirin is an irreversible inhibitor of the enzyme called cyclooxygenase, or COX, cyclooxygenase. Basically, the aspirin will enter the active site of the enzyme, reacts with the serine hydroxyl group. Basically, it adds an acetyl group, or it acetylates the serine hydroxyl, and that inactivates the enzyme. It basically kills the enzyme cyclooxygenase. We discussed icosanoids such as prostaglandins in a previous video on lipids. Cyclooxygenase is an enzyme that is required for the synthesis of prostaglandins. So once this enzyme is inactivated or inhibited, there is decreased prostaglandin synthesis. And many of these prostaglandins are very important for things like inflammation, swelling, pain, and fever. So by inhibiting this enzyme, we decrease the prostaglandin synthesis, and therefore we can control, or we can reduce the inflammation or the swelling or the pain or the fever. So aspirin is an example of a non-steroidal anti-inflammatory drug, or NSAID. Let's look at another example of irreversible inhibitor. And in this case, we'll look at the inhibitor or the drug called 5-fluorouracil or 5-FU. This drug is what we call a mechanism-based inhibitor, also sometimes called a suicide substrate. So let's look at how this drug works as a suicide substrate. So the drug itself, 5-FU, is not an inhibitor. However, when we take in the drug, this drug is converted, the 5-FU is converted into FDUMP, so fluorinated form of DUMP. Now DUMP is the substrate for the enzyme thymidylate synthase. This enzyme converts DUMP into DTMP, and this DTMP eventually will get converted into DTTP. Now DTTP is a substrate required for the synthesis of DNA. Okay, and of course DNA synthesis is very important for dividing cells. Cells that are actively dividing, they need to make a lot of DTTP because they need to copy all their chromosomes. They need to copy all their DNA. So when the fluorinated form of DUMP, so instead of DUMP, if we have the fluorinated form of DUMP, if this goes to the active site of thymidylate synthase, it will covalently bind thymidylate synthase. Okay, so the FDUMP will be covalently bound to thymidylate synthase, and that will essentially kill this enzyme. And without this enzyme, DTMP will not be made, and without DTMP, there won't be enough DTTP, and this cell that is actively dividing, for example, cancer cells, that cell will not be able to make new DNA, and therefore it cannot divide. So this is a drug that is used as part of cancer chemotherapy. Okay, so now let's look at reversible inhibitors. So these are inhibitors that only temporarily diminish or reduce the enzyme function. There are three major types of reversible inhibitors. In the first type, what we call competitive inhibitor, a competitive inhibitor binds only the enzyme, the free enzyme. That means the enzyme doesn't have a substrate bound to it. That's called the free enzyme. An uncompetitive inhibitor will only bind the enzyme substrate complex. That means the enzyme already has a substrate bound, and now the uncompetitive inhibitor will bind. And in the third type, what we call mixed inhibitor, a mixed inhibitor can bind the free enzyme and the enzyme substrate complex. Okay, so we have three types: competitive, uncompetitive, and mixed inhibitor. So remember, reversible inhibitors are very important for the human body because this is how we can regulate many of our enzymes. That's like reversible inhibition. And by regulating the enzymes, we can regulate the reactions that are happening in physiology. In future videos under metabolism, we shall look at the different types of enzymes, the different classes of enzymes. The most commonly inhibited enzymes are the transferases, the oxidoreductases, followed by the hydrolysis. Then the lyases, ligases, and isomerases are also inhibited to a lesser extent. The most common type of inhibition we see in the body is competitive. That's about 75%. Non-competitive is only about 18%, And uncompetitive inhibition in the body is quite rare. It's about 7%. The most inhibited pathways include glycolysis, which is the breakdown of glucose, gluconeogenesis, which is the synthesis of glucose. So clearly glucose plays a very important role in the human body, and therefore it's not surprising the enzymes that are involved in glucose metabolism are very, very highly regulated. So besides glucose metabolism, nucleotide interconversion, the enzymes involved in nucleotide interconversion are also very finely regulated. That's not surprising because nucleotides are necessary for making your DNA and your RNA. This information is from this publication in Nature Communication, which also showed that by putting the enzymes in compartments such as your mitochondria or your peroxisomes or your ER, by subcellular compartmentalization, the cell can reduce enzyme inhibition. So let's look a little bit at these major forms of inhibition: competitive, uncompetitive, non-competitive. Now, non-competitive is a type of mixed inhibition, non-competitive, and then we have uncompetitive. So competitive inhibitor. Now, competitive inhibitor is the most common way to regulate enzymes in the human body, and it's also very common in pharmacology. A lot of drugs actually work as competitive inhibitors. So competitive inhibitor competes with the substrate for the active site. So the inhibitor and the substrate are both competing to bind at the same site, that is the active site of the enzyme. So it's not surprising that the inhibitor has a chemical structure very similar to the substrate. So here's an example. The example here is for the enzyme succinate dehydrogenase. This is an enzyme we'll talk about under metabolism in the TCA cycle in a future video. Succinate dehydrogenase has the substrate succinate, shown here. The inhibitor of this enzyme is malonate. If you look at the structure of malonate and succinate, you see the structure is very similar. Malonate has one less carbon compared to succinate. So malonate competes with succinate at the active site of the enzyme succinate dehydrogenase, and therefore malonate is a competitive inhibitor of SDH, succinate dehydrogenase. There are also many drugs that work as competitive inhibitors. Here are two examples. Methotrexate is a competitive inhibitor for the enzyme dihydrofolate reductase that we'll talk about under nucleotide metabolism. And lovastatin, it's a statin drug that's reduced to lower blood cholesterol, and we'll talk about this under cholesterol synthesis. This drug is a competitive inhibitor to the enzyme HMG-CoA reductase that is very important in controlling how fast our body synthesizes cholesterol. Uncompetitive inhibitor, again, very rare in the body, very rare in the body. It is an inhibitor that will bind only after the substrate binds to the enzyme, because it binds the ES complex. Okay, so once we have the enzyme substrate, only then will the inhibitor bind to the ES complex. Now, this uncompetitive inhibitor does not compete with the substrate for the active site, so it does not bind at the active site. Instead, it binds at another site on the enzyme, And we call this other site, which is not the active site, we call this the allosteric site. So it binds at the allosteric site. There is no structural similarity between the inhibitor and the substrate in case of uncompetitive inhibitors. So although we don't need the inhibitor and the substrate to resemble one another in terms of chemical structure, like we saw in malonate and succinate, like we saw here for competitive inhibition. But it's interesting that studies have shown that in the body, about 31% of the uncompetitive inhibitors actually do have a structure quite similar to the substrate. Examples of uncompetitive inhibitors, again, this is very rare. One of the best examples is L-phenylalanine is a uncompetitive inhibitor of the enzyme alkaline phosphatase that is produced by the intestines. Okay, so this enzyme is produced by different organs, including bone and intestine and so on. L-phenylalanine is a uncompetitive inhibitor only for the intestinal alkaline phosphatase. Example of a drug, valproic acid is a drug that works as an uncompetitive inhibitor for an enzyme that's very important in liver metabolism of drugs, and that is UDP glucuronide transferase. And we'll talk about this enzyme later on under metabolism. And the third type of inhibitor is called the mixed inhibitor. So this inhibitor binds both the enzyme as well as the enzyme substrate. So since it can bind the enzyme and the enzyme substrate, it can bind enzyme and enzyme substrate. So in a way, we have three types of mixed inhibitors. So in one type, this binding is much stronger to the enzyme, free enzyme, compared to the enzyme substrate. So this is much stronger than this one here. In the second type, this one is much stronger than this one. That means the inhibitor binds ES much stronger than it can bind E. And in the third type, the binding of the inhibitor to the free enzyme is the same as the binding of the inhibitor to the enzyme substrate. So if the inhibitor has equal affinity to the free enzyme and the enzyme substrate, then we call that type of mixed inhibition a non-competitive inhibition, or the inhibitor is a non-competitive inhibitor. So remember, after competitive inhibition, a non-competitive inhibitor is the second most common type of inhibition that we see in the human body. This non-competitive or mixed inhibitor, just like uncompetitive inhibitor, binds at an allosteric site. It does not bind at the active site, it binds at the allosteric site. Once again, we don't need to have a structural resemblance between the inhibitor and substrate. So although we don't need this, interestingly, 1/3 of non-competitive inhibitors that have been studied have a structure that is quite similar to the substrate. Some examples of non-competitive inhibitors, alanine and ATP, these inhibit pyruvate kinase. This is an enzyme in glycolysis, and this inhibition is non-competitive. Malonate inhibits aldehydes. Malonate is a non-competitive inhibitor of the enzyme lactate dehydrogenase. The drug mycophenolic acid, a very important drug for immunosuppression. This drug is a non-competitive inhibitor for the enzyme inosine monophosphate dehydrogenase, IMP dehydrogenase. Cyanide is a mixed or non-competitive inhibitor of the enzyme cytochrome C oxidase, which is very, very important in the eventual synthesis of ATP in the mitochondria. And then heavy metals such as lead, they are mixed or non-competitive inhibitors for many, many enzymes in the human body. So this is an introduction to inhibitors, irreversible and reversible inhibitors. And in the next video, we will see how these reversible inhibitors that are very important for regulating enzymes in the body, how do these reversible inhibitors affect the Vmax as well as the Km values. So until the next video, goodbye for now and have a wonderful day.